Normally we can do very a lot of things, mental functions all normally, but in schizophrenia patients they have a problem with. The symptoms of schizophrenia patients include three main. Here is actually spying on me and reporting to the Prime Minister. Some weird kind of unbelievable kind of false beliefs. Able to enjoy life, not able to talk easily, not able to feel pleasure of living, all these things. So this is like they're called negative symptoms. Then there is a third category of symptoms called cognitive deficits, right? So these people when they want to, like if you do, you and I do like a small cognitive deficits, right? So these people when they want to, like if you do, you and I do like a small test, and will score reasonably well, whatever like significantly lower than them. So they have all these buckets of symptoms. Now, unfortunately, the first drug discovered and used for schizophrenia. I'm sorry? Antipsychotic. I can't hear what. Yes, atypical and typical. What category, what pharmacological category of drugs are they? What kind of receptors? Dopaminergic, right? D2 receptor, dopamine, and that is what they are used for. Now, discovered long and it works. They work very well, reasonably well against positive symptoms, that is delusions and hallucinations, but they cannot do anything for negative symptoms and for cognitive symptoms. Okay, that is the problem. And unfortunately, since about almost 70 years, there have been no new drugs. They have been typical and atypical antipsychotics. They are just marginally different. If you look at all the high-level outcomes, you know how much better each drug performs. Basically, they're very good in stopping positive symptoms, but they're not very good for negative symptoms and cognitive symptoms. Now, obviously, there's a huge burden of schizophrenia. People, about 1% everywhere, whether it's in the US or India or somewhere in China, everyone going through all kinds of mental problems. A lot of them succumb to things like suicide. So almost if you compare the people with schizophrenia, without schizophrenia, and you look at their lifespan, almost 25 years difference between those. So it's very sad. You know, young people, if they're not only suffering through it, but also end their life prematurely. It's very devastating not only for them, but also for their families and friends. So that is why there's a huge need for good drugs that can not only address the positive symptoms, but also address the negative symptoms and cognitive symptoms. So that is what we call as unmet need. So there's a significant unmet need, not only in the area of schizophrenia, but also in depression and all other mental illnesses. Okay. Now, one of the problems with this antihistaminic drug, one of the histamine on it, it contracts, and then you give the drug. If it relaxes it, it's an antihistaminic, and then you develop a drug like that, right? Unfortunately, for mental illnesses, because the functional problem is about mental function, you can't replicate hallucinations and delusions or negative symptoms easily in animals, right? Because animals don't think like us; they're very, restricted in how they're thinking and all that. So, for a long time, people used to say they would suspend a mouse like by its tail like this, and the mouse will keep on struggling like this. I mean, try to like write its posture and all. And they would say, okay, this is a model of depression, right? And then they would give some drugs, and if they stop struggling, maybe because its muscles have relaxed, they would say, oh, this is an antidepressant. So there's models like that because of mental connection. You can't really easily replicate human conditions like depression in animals. A lot of times people would discover molecules that would work very well in the animal models, go to the clinic, put a lot of money in it because clinical trials are expensive, right, as we know. But once they do that, the drugs would not work. Like this, again and again, there have been a lot of failures. And so there's a big need. Now, NIMH is NIMH is National Institute of Mental Health. It's a big organization in the US that basically supports mental health research. They said, you don't worry about models because you really can't replicate human states in animals, mental states. So instead, focus on what are called biomarkers that are more functional, that is functional to make the brain work better. Okay, how the brain circuits, the neurons together are working. Use that because whether an animal is a mouse or a human being, basic neural processing is the same. What we do with that information is different. For example, you and I come into a new room and you see a lot of people and you're checking them out with your eyes, right? You're looking at them. A mouse, when it comes into a new place, what does it do? It will move its vibrissae, that is its vibrissae that are going across from her mouth. She will move that and then feel everything. She will smell it. Essentially, it's doing the same thing. We are looking through vision and it is processing through feeling and then through smell. So even though those behaviors are comparable, they are not the same. So that's why don't focus on behavior. Instead, focus on the network activity. In this case, the vibrissae network activity will show some response. Similarly, when we are looking at maybe our visual cortex is activated, so there is some kind of a correlation there. So that is why there is increased push to look at circuits rather than behavior. Is that clear? Yes? Okay. So behavior is not easy to replicate in lower animals, whereas neural activity, it's very easy to replicate. So I'm going to talk about one particular kind of neural response called EEG. Have you heard of EEG? Basically what? You put electrodes of the brain. It's no different from, say, if you put leads on your hands and then you can record the QRST waves, right? The heart activity. Similarly, every time the neurons are firing, they contribute a small potential to the brain. If you put electrodes on the brain, you can overall record the potential of the brain. And typically that response looks like an oscillating response, right? Then the oscillations tend to be of various frequencies. Delta, that is 3 or 4 hertz or 3 or 4 cycles per second. Theta, 6 to 9 hertz per second. Alpha, so on and so forth. Alpha, beta, gamma. I somehow, through my research, I was focused on gamma activity. That is, gamma activity is any kind of oscillations that happen between 30 to 100. Talking about something gamma frequency and the high level that focus on gamma oscillations is in our brain, different regions tend to see with something called alpha. So if you put electrodes here, you will see 9 to 10, 10 to 11 oscillations like this, like this, like this. And then it's very clear if you close your eyes. But the moment you open something, those oscillations go down. Okay, so that is peculiar to the occipital cortex or where our vision is processed. In the prefrontal cortex, where this region is called the region that takes care of executive function. What is executive function? Executive function is to make decisions. For example, I want to go to seminar, I have a class. Which one would I go? I say, class, tomorrow I have exam. This class is not in that portion of the exam, so it's okay to miss. Something like instead of the seminar, right? So things all the time, decision making, okay, which should I follow? Should I listen to this person in a traffic or should I listen to somebody else? That focusing attention, all that is a part of what is called executive function, and this around here. And in prefrontal cortex, typically when you're thinking a lot, you're mentally involved, you will have what are called gamma oscillations that are like 30 to 40 to 50 hertz per second, that is cycles per second. So we thought that it makes sense to focus on this because typically in schizophrenia patients, the region that seems to be most affected is this. They execute, attend to things. They want to say, okay, as you're listening to, for example, say I'm talking and you're listening to it, they can't keep the attention. We can keep, right? Most of the times we can keep attention. The fans are spinning, we can hear them, but we can just push them away, right? Because you're attending on this speech, that becomes deprioritized. That is because of our executive function. people who don't have this function, they will get distracted. Oh, fan is spinning. And you sit and then suddenly you'll feel the chair is soft, hard, whatever. So you can't keep your attention focused on one thing. That is a function of our executive function, and schizophrenia patients tend to have a lot of problems with that. So I talk about a biomarker that is called 40 Hertz auditory stimulation steady state response, and it's a pharmacodynamic biomarker. What does pharmacodynamic biomarker means? Yes? Yes, mechanism, and also mechanism based, correct? And also it's a marker that responds to pharmacological treatment, right? It may not actually show you whether the person has cognitive deficits or not. That is not what it is. What it tells you is if you give a drug, whether it can increase it or decrease it or improve it and so on. So that's what. And that's what I'm interested in, or those of us who are interested in pharmacology should be concerned, because when you're trying to develop drugs, you need good markers that are translatable, that is, you do it in animals and you take the same marker to the humans, you test again. So that's a translation should be there. So those are conditions of good pharmacodynamic markers. What are biomarkers? Many of you have already answered what biomarkers are. In this case, we are focused on CNS-based biomarkers. Next slide, please. Okay, now, why are biomarkers critical for development? So there are a number of reasons and they're listed here. Preclinical drug optimization. For example, if you are working in a department, we'll make a dozen compounds. Okay, that will be in first 10 days. If they're very productive, another month, next month, they'll make another dozen compounds. Right? How do you choose? You can only choose one or two to test in clinic, right? Ultimately, that's your goal. But it's not easy to prioritize them. How do you prioritize them? You prioritize them based on in vitro assays, and if they work out, let's say if you're developing a drug for dopaminergic, whatever activity, then you do in vitro tests, and once you find something that has very good efficacy, that means it has high affinity to the receptors and it has a very good intrinsic activity, then you will go and look at other kind of tests where it's not binding to other receptors that you don't want it to bind, right? Then once you identify those molecules, for example, in rodents in this case. So that is one reason why you need good biomarker. Two, dose selection and functional engagement in phase 1. So for example, you have a good drug. How do you know which dose to test in human patients? Right? It's a new drug. We have no information. How do you do it? You do it because using a biomarker. If you think that the prefrontal cortex circuitry is important, then you use some task that engages this and see if your drug in these healthy volunteers is enough or not to change that. Then you take a range around that value and test it in patients or in a larger sample. So that's how you do it. So that is called dose selection and functional engagement. What is functional engagement? Functional engagement is whether the drug is binding to its target and showing some kind of functional response. Right? In humans, for example, you can't take their brain and look at receptor binding. Right? That's not possible. So you have to infer from the data you have. You do all that in animals and then you have this data, then you say, okay, if this works, it should increase, let's say, cortical activity in the brain, right? You can say that. Then you look at the drug in the clinical trial, see whether it is doing that, then you say, yes, target engagement is there. Let's say you did not have that and you proceeded to do your clinical trial. If there is no response, then is it because the drug never went into the brain, or is it because it is ineffective? You can't answer that question. If you can't answer that question, having spent millions of dollars or millions of rupees, basically you wasted companies' money and your own time. Right? Process using solid empirical data. That way we use that in this case. Next, patient stratification. All kinds of clinical trials. Some of you found these industry. These are the terminology you'll use, whether it is cancer or CNS or whatever thing. So these are all the same kind of principles. So in the other thing is that all our populations are not distributed, right? Our heights are all normally distributed, right? Whatever, our weights are normally distributed. In no way are we all the same, right? So what does that means is that is because of genetic heterogeneity. But at the same time, if you really look at it, there is also a lot of small difference. when you are doing a clinical trial, in order for you to improve your chances of success, you want to identify those people who are going to respond and avoid those people who are not responding. Right? That is the basis. That is another where you do patient stratification. For example, if you take a schizophrenia patients, normally the 40 hertz response is small in them relative to the control. But everybody's response is not going to be small. Some may have good response, others may have bad response. If your drug is going to improve that response, you cannot take the patients with good response and say, I'll improve it. It's hard, right? You may run into the ceiling. So then what you do is only norm is lower than the regular controls. That is patient stratification, or sometimes it's called patient enrichment. You are enriching your sample with likely people who are going to benefit. Last is prediction. Prediction is essentially saying, okay, if this 40 hertz performance, something like that. So it's not necessary by biomarkers, but a lot of times you try to make sure that you can at least get some information that's valuable in any of these categories. Okay, so this is just a slide reminder saying that because of the CNS diseases, tissue sampling and all from the brain, you need good biomarkers, functional biomarkers that can address a lot of these diseases, including schizophrenia, Alzheimer's. Okay, so this comes back to what I was telling about, because models cannot be the same as human behavior and all. Instead, focus on functional circuits. In this case, something a mouse or a rat may do it with their vibrissae rate. That activity in the somatosensory cortex for rat may be equivalent to some of us processing vision through visual information. So that is what they're saying when you say use equivalent functional circuits that tell you something about the region in the brain. Okay, so this is essentially saying this. You can look at problems in many number of ways. You can look at it on the behavior, which is there, the cats, interacting with each other. That's an example of behavior. Then if you go back, you can look at the genes, and then you can look at neurons, right? All of these are valid ways to look at a problem. For me, this is more interesting because it's both functional and I don't have to destroy the system. I can just use the system as is and look at the response and then use the same thing in clinical populations. Okay, next slide please. Okay, so EEG, as I said, is very interesting because it's the activity of the brain. You can pick it up. You can pick it up and you can look at the response, and each of those, the nice thing about EEG is if you look at this graphic on the left side, you may not see it very clearly because this thing, but basically there are lower animals like mice and rats at the bottom and humans at the top. Throughout the evolutionary tree, these responses are essentially conserved. What does that mean? That means essentially the behavior of this EEG tends to be very similar across species. Now, anytime there is a conservation, what does it tell us? It tells us that that system is extremely important for survival. Because if it is not important, then it would be eliminated, right? Over thousands and millions of years, if some physiological phenomena is preserved, that means it's very important for evolution. So this is a very evolutionary conserved response. And the advantage there is you can study in rats, you can study in mice, you can study in monkeys, and you can study in humans. It tends to be very similar. So the other thing is patterns reflect brain state. So whether it's a rat or a human or a monkey, if an animal is sleeping, you will have 3 to 4 oscillations per second in the brain. Okay, that is very well known. And similarly, if the animal is very, it can escape, it's trying very hard to look for the window. In those situations, you'll have a lot of beta and gamma oscillations in the front. Similarly, if you ask somebody to solve a puzzle in humans, where they're trying to solve a problem, then your beta and gamma oscillations are very high. So it's again functionally conserved across species. And the nice thing, as I'll show you also later on, is these responses tend to be, they tend to be responding other thing that is very interesting is when an oscillation goes on like this, up and down, it has some nice physiological significance. Essentially, it's a reflection of how, how you've studied action potentials in physiology, right? Resting membrane potential, hyperpolarization, then some stimulus and depolarization, right? And then comes back and hyperpolarization, and then comes back to resting membrane potential, right? So basically, this is a reflection of that resting membrane potential, not in one neuron, but billions of neurons. That's what it is reflecting. What that means is that just like a neuron, if you stimulate it when the resting membrane potential is low or hyperpolarized, it's very hard almost at the resting membrane potential. Then you stimulate it, it's very easy to make an action potential. Similarly, these are like that. If there is up and down oscillations, at one point if you stimulate, let's say at the top of this line here, then the response tends to be larger. Okay? It is at the bottom, at the trough level, response. And this is the way the physiological system our brain optimizes. That means not always active, want to conserve resources, right? Because mitochondria is there, ATP is being produced, it's trying to use up all the energy, it doesn't want to waste its energy. So it will do it only in like little snapshots. And a lot of times when you have rhythmic entrainment in the response, all the brain is doing is it's trying to optimize the way it can react while taking rest in between. That's what is happening. So that is one physiological importance of studying oscillations, because it reflects the way the brain optimizes responses. Okay? All right, next, yeah. So here is an example where these oscillations are reset. What does that mean? That means in the left side, when they say trials, you can look at the oscillations. The first, second, third, fourth, fifth, they're all different trials, right? They're testing the brain's oscillations. When there is no stimulus, they're all randomly oscillating, right? There's no pattern here. It's all, one is high, one is low, and so on. But once speech starts, somebody starts speaking, then all of them starts oscillating in the same way. This is called phase reset. So phase is going up, going down, up, down. This is the phase. And the reset is when it all lines up properly. That is phase reset. Now, turns out that when we are talking, when you're listening to somebody, there is a rhythm to your speech. There is about 3 to 4 cycles per second. That's how we speak. We don't speak continuously. Human speech is always rhythmic. Even natural sounds, animal cries, they're all rhythmic. And that rhythm is important because once I start speaking, or once you're speaking and I'm listening, my brain suddenly resets. It resets and starts oscillating at the frequency of your speech. And how does that help? That helps us to focus, because you're not wasting your energy continuously, but rather, okay, the sound is going to come 3 or 4 times a second. I'm going to optimize my resources to match that. Okay, that's called resetting. And another example is if you're a marching band, if somebody is marching left, right, and you have missed a step, right? What do? You wait and then you go again, right? That is the resetting part. That is what happening here, resetting. Okay, next slide, please. You can skip this. All right. So the particular pharmacodynamic biomarker I am going to talk about, and that in which I am very interested, is called a 40 Hertz steady state response. That's a long name, but all it is simply click train, right? Clicks, 1, 2, 3, 4, like that, clicks. And they go 40 times a second. Okay, it sounds somewhat like this, like that. So you do multiple times. You get, that's one trial, 2 trials like that. So you do multiple trials like that, and then you average them and you see the response. So when you do that, you see a nice response like that, as shown in the graph. And then again, this it's not very bright, but basically what you see, you get a nice, very clear. Next slide, please. Okay, so the reason this is interesting, particularly in the context of schizophrenia, is that normal people will show a nice response. Schizophrenia patients show a weak response. So the synchrony is strong and control relatively speaking, it will be strong, and in schizophrenia patients, it tends to be weak. Next slide, please. Okay, so the other thing is, now that's okay, that's just an observation. What has that got to do with our cognitive functions? Because it's not really, in cognition, you're not really looking at 40 hertz click trains and looking at the response, right? You're trying to solve problems, you're trying to answer questions and also that is cognition. Turns out that there is a nice correlation, there is a lot on this slide, but I'm just saying this very simply. There's a nice correlation between 40 hertz auditory steady state response synchrony and your ability to perform well in some cognitive tests. Right? What does that mean? That means because it's a linear correlation, you can say that okay, if somebody is deficient in 40 hertz responses in entraining them, then chances are that they won't perform very well in a cognitive or of correlations. Yes? So there is a simple correlation. Auditory steady state response is cognition, it's just a response, it's just a neural response. Turns out that the neurons that don't do a good job in responding to it also seem to not do a good job in processing cognitive tasks. Okay, that's the relation. Okay. Now can we go to the next slide, please? Okay. So again, we're coming back, right? Because I'm a pharmacologist, I'm a pharmacodynamic, I'm interested in pharmacodynamic assays that you can use to prioritize drugs so that we can develop new drugs for patients. I wanted to see whether I can develop the same thing in rats. So this was a long time back we did this work. Basically for rodents, if you take the frontal or even from auditory cortex, you can record responses, play these click trains one at a time and then average them. You see a nice response like this. This is a response of the EEG because what happens is the EEG normally looks like this, right? Oscillations. But then you start playing this, everything gets synchronized, right? And they all start oscillating. Then what happens is when you take many trials of it and average them, what happens is the random oscillations will become smaller and smaller because you're combining them together. Right? Makes sense? Random ones get smaller, the time locked response becomes stable and they will start showing up. So they become more and more obvious. So that's a lot of power in averaging over a number of trials. Your random response gets smaller, your time locked response to the click train becomes larger. So what we call that as a high favorable signal to noise ratio. Something that you will look in all kinds of assays where you want the signal to be high and the noise to be low. Have you heard of signal noise ratio? No. But in any assay you do, whether it is in pharmacology, medicinal chemistry, whatever it is, pharmacognosy, whatever it is, when you have a response that you are interested in and the background activity, there should be a large separation. Only then you something like, for example, if the fever or consistently having 99 degrees, you won't be able to tell because there is no difference between the signal and the noise, like that. On the other hand, if it's 103 and 99, then you can tell, right? So signal noise ratio is something that we look for all kinds of assays. Some of you might do PhDs later or M.Pharm and you may do projects. Anything you study, you want it to be like a larger gap between background and the signal you're interested in. Okay, so this lower panel is just showing that means there are all kinds of frequencies in it, but if you use a filter, band pass filter, you can just isolate 40 hertz because the clicks are in 40, you can isolate that signal by filtering it between 38 hertz and 42 hertz. So you can narrow band, you get a nice response like this. And this essentially is saying that rats also behave like humans in the context of this 40 hertz auditory steady state. Okay, that's all this is. Okay, next slide please. Okay, then the other thing is assay, how good your assay is. Now, it's one thing for you to do some kind of assay, but in drug development, what you do is you do again and again and again the same response, the same assay. Okay, one batch of drugs, like I said, somebody gave me 12 drugs, I evaluated that batch. Next batch, I do the same thing. Next month, I do the same thing, right? But what if our assay changes every day? Can I compare these drugs with those drugs? No, because we don't know whether they are comparable or not. So what we need to do, particularly in drug development, is that to make sure that your response again and again and again stays the same. A lot of people don't worry about this, but actually a good scientist make sure that the experiment that you are doing is reproducible across times. Then you can be say you can say this assay is very good. We can use we can use this to screen drugs. So that across 0, 30, 60, 90, 120 performs more or less the same. It's not exact. It's more or less the same. And each of these lines is every week he was testing it twice. So he was doing this twice every week, and he did it for like 8 weeks or 6 weeks, I can't remember, but basically again and again. The bottom line is it had very acceptable test, retest reliability. Have you heard of test, retest reliability? So those of you who are doing PharmD, you may have heard anything that you study should have test, retest reliability. So that's what this is showing. Next slide. Okay, so coming to mechanistic. So we showed that rodents synchronize to 40 hertz click trains. What is the mechanism behind? Because that is important, because if you want to discover new drugs, let's say you want to discover new drugs that are cholinergic, right? You need to know that cholinergic drugs will have an effect on the system. You can't expect every drug to be effective on this, right? If cholinergic pharmacology is involved, then it should have a response, like that. One of the things in schizophrenia area that's very big is use of, there is this hypothesis. In schizophrenia, the original hypothesis is that dopamine is very high in the subcortical regions, in the striatum, and they have hallucinations, and it is very less in the prefrontal cortex, reduced. But it cannot do much. If anything, it will, because the activity is already low in the prefrontal cortex and it's further. Then your activity is even reduced. So that's it. And delusions. Make sense? Okay, so that's what dopaminergic hypothesis does. Now, there is a competing hypothesis called glutamate hypothesis. What does glutamate hypothesis say? Glutamate hypothesis says that glutamate acts on receptors. Yes? Yes. Very important for memory, for example. Glutamate acting on the NMDA receptors makes your prefrontal cortical circuits work well, and it also in the striatum suppresses the dopaminergic neurons from becoming hyperactive. Okay? How did the proof for this hypothesis come about? In the 1950s and 1960s, there was a street drug called PCP, phencyclidine phosphate. So people would abuse it. They would like snort it, or they would use it, and then suddenly they would become like schizophrenia patients. So they were brought to hospital. This guy is schizophrenic. So no, he's not schizophrenic. He was normal until yesterday. Then what happened? They used PCP. Happened. They discovered this connection. PCP is an NMDA antagonist. Therefore, NMDA antagonism may be playing a role in causing them schizophrenia symptoms. Therefore, they came up with this hypothesis that NMDA receptors are involved in schizophrenia. So it's I'm simplifying it, there's a lot more evidence, but nevertheless it's a hypothesis, and there is a big area in this, in drug discovery, which are focusing on NMDA activity to see if we can produce good drugs. The problem is, NMDA, what kind of transmitter is it? Is it excitatory or inhibitory? Excitatory, right? And then the more it is there, it can cause cellular death also. So it has to be very precise. It should come and go. If it's not long, it can kill the cells. So the NMDA thing is that you have to test it in a way that you cannot do too much of the effect. It has to be very regulated. So this is the hypothesis we tried to study in rodents, because it was not studied in humans. It's not easy to give experimental drugs like NMDA. Other than ketamine, you can't give it to humans. So we tested in animals. Next slide, please. Okay. Here is again, the slide is not completely clear, but let me tell you what is happening. These are normal animals treated with vehicle, and this is the noise level. Okay, then the click train starts here. It says like that. It goes on for half a second here. So when it happens, nothing happens, and suddenly here, look what is happening. The neurons are all, the field potential suddenly begins to oscillate. You can see that, right? It's becoming more organized. Here there is no pattern. Here they're suddenly organized and they're responding to the clicks, right? This is the phase resetting I talked. So they're walking and then suddenly they start marching at 40. So for that it takes about 250 milliseconds. 250 milliseconds is what? It's 1/4 the time of 1 second, right? In that is the time it requires for them to start marching to the beat. Okay? Now, next question is, because we were interested in NMDA mechanism, what should NMDA antagonist should do to this? It should reduce it, correct? Yes, it should reduce it. So that's what we tried. Now we gave, in the same set of animals, we gave NMDA antagonist and looked at the response. Now, look what is happening. It is showing some kind of response, but it's not the same as this. Here it's much more organized, here it is disorganized. So, signal, noise have become more closer than before. Before, signal was this, noise was this. Look what happened to the noise. The background has become actually wider. So the NMDA has made the mouse more like a schizophrenic mouse. Or that, right? Its brain processing has become worse in the background. Then the signal is much less. So the signal noise has come down when I gave them the drug, NMDA antagonist. Okay? Next slide, please. Okay, so this is another paper from another paper, data from another paper. It's just showing that when you give vehicle, you get a nice, this big yellow band is essentially the power or the signal power of the 40 Hertz response, and then in the presence of MK-tone, it goes down. And similarly, that synchrony, every time there is a click, how well is it organizing? Similarly, that is this, and that is also reduced by MK-tone. So this kind of work is called validation. If you have a hypothesis, you have to make sure that your model is showing the same thing, right? If your hypothesis and NMDA antagonist will reduce the effectiveness of the function, brain's function, then you have to show that it is. When you test it in animals, you should show that. That's what we are doing here. We are validating the model. Okay, we are showing that MK-tone, which is an NMDA antagonist, blocks this response. It blocks its power. It blocks its synchrony. And like I showed earlier in the previous slide, it also reduces the signal noise characteristic. Now, you can imagine in schizophrenia patients, they are not deaf. They can hear all these things. What is the difference? The difference is their noise tends to be, background noise tends to be larger and their signal tends to be lower. So as a result, a lot of things we process, they can't process very well. They will hear it, but they need higher threshold, everything has to be calm for them to hear and all. Otherwise they'll ignore it. That is the problem. And it seems like in this very limited way, this kind of model also reflects that kind of behavior. Is this making sense to you all? Huh? If you have, like even if it is, 3/4 of it you're able to understand, which is good, because this kind of knowledge you can apply in other situations also. How to process, information, and especially if you're doing PharmD. Okay, move on, next slide please. Okay, so this is just sort of data showing that nicotine. Now, nicotine we know is not a good drug. It's it's present in cigarettes. The idea is not that nicotine will be used as a drug, but nicotine has some good characteristics where it can actually improve cholinergic system and make you a little bit perform better. Now, if you go a little higher, then your performance falls down. So it's called an inverted U-shaped response. A little bit, performance improves. A little more, the performance falls down. Okay, that's the characteristic of nicotine. So we just wanted to see what will a cholinergic agonist do. Instead of doing something else, we just looked at nicotine. What we showed was nicotine nicely improves the response. So you can see how this is light yellow, relatively speaking, and this is dark yellow, right? It has improved the performance of the steady state response. Okay, now interestingly, we wanted to see, okay, MK-tone, that is NMDA antagonist, blocks the response. Nicotine improves the response. So what does that tell you? That this is a pharmacodynamic assay, right? Why? Because you can reduce it and you can improve it. Then you have that range. Then those kinds of assays are the ones that are suitable for clinical trials where you can improve and reduce. You should be able to see the dynamic range. And pharmacologists like me love this kind of range in response. When you have a low response, high response, medium response. Okay, so there's a lot more work we did, but I'll just shorten it for you. This is clozapine. What kind of a drug is clozapine? What is it? Atypical antagonist. Very good. So it's a D2 antagonist, but it's also atypical antagonist. Do you know the oldest atypical antagonist? Yes, you have olanzapine, you have risperidone, you have all sorts of drugs, right? So what is the oldest atypical drug? There was a study in Norway where a whole bunch of died because of clozapine. And what pain does is it can cause bone marrow depression. What does that mean? It can bring your white blood cells to very low levels, like granulocytosis. And so they got very scared about it and stopped using it. For a long time that went on. Later on it came back, they started saying, okay, if you want to use clozapine, you have to monitor the blood counts. See if they have a granulocytosis, then you take it away. If they don't have a granulocytosis, because not everybody has it, it's an atypical response, it's an idiosyncratic response. So not everybody has it, and when they don't have it, then it's okay to use. Okay? But the nice thing about you can take all the drugs, all the antipsychotics you have, you keep them in one bucket, and clozapine in another bucket. Why? Because if you look at outcomes in cognition, in schizophrenia symptoms, so many other things, clozapine tends to be better than all other ones are in one bucket, clozapine tends to be in another bucket. Now if you look at it pharmacologically, it's a very difficult drug in the sense you can't be sure about what its mechanism is. Now for all antipsychotics they are D2 antagonists. Clozapine is also a D2 antagonist, but it's also an alpha agonist, it's an alpha antagonist, a muscarinic antagonist, all kinds, you name it, it will be there. There will be like 15 different receptors to which it binds. So mechanistically we don't know. People have spent a lot of time and energy and money trying to discover what clozapine is so that they can make billions with a new drug, but they could not because it's very mixed, what we call a dirty drug. It can bind to many targets. So without going into its mechanism, we tested clozapine, and clozapine had a beautiful dose-dependent increase in phase synchrony of the 40 Hz response. So it made those gamma oscillations a lot more, nicer, more stronger, more aligned stimulus. That's what clozapine did. So we were very excited about this. This was work published in Schizophrenia Bulletin, which is a very good schizophrenia-related journal, and this was a good study. Next slide, please. So we looked more closely what clozapine is doing. Is it just improving the synchrony, or is it doing something else? So that's what is shown here. This is just a normal background of multiple trials combined together, and then this is the spot where that click train, that thing gets turned on, right? It goes on all the way here, and then we are looking at the same group of animals, how they are responding in vehicle condition, in haloperidol condition, in clozapine condition. Okay. Only one dose is shown, a small dose of clozapine is shown. Look what is happening. It takes about 200 milliseconds for the vehicle group control animals to start becoming synchronized. Okay, then they're oscillating and synchrony. Until then, they are just random. Look at what clozapine does. Almost 125 milliseconds before, they all start oscillating at the same time. So clozapine accelerates the phase synchrony. Right? It makes the brain do its work faster and better. In this case, I don't mean to say it will make it for everything, but in this assay, it is doing it. So it's a remarkable thing, and this is also the first time anybody showed that this phase synchrony can be actually brought forward using a drug. So now, because we published this, I'm just sharing this because you'll understand if you're a pharmacist or you're a pharmacologist or you're in this line of work, how you use this information. We published this, and then several companies that are developing new drugs, they contacted us and say, hey, can you test if it works or not? one company's drug showed nothing, and another same kind of response, and it was a muscarinic receptor agonist. So we were able to show. Now we are testing whether the antagonist of that muscarinic receptor will block clozapine's effect. So that's an assay we haven't done yet, but we'll be doing once I go back. Next slide. So yeah, this is a summary. I shared with you a model that we developed based on the 40 Hz steady state synchrony response that you see in humans. It's a general pitch that electrophysiology based models tend to be good. They tend to be translational. You can use it in animals and you can use it in humans. It's a nice, and you don't have to like poke them or you you don't have to take any tissue samples and all. It's a nice non-destructive way to study the brain. And generally speaking, this 40 Hz response is responsive to both an NDA mechanism and even perhaps cholinergic mechanisms. So with that, I conclude. I'm very happy to answer any questions or you have any doubts about this or something else about industrial drug development, drug discovery. I'm happy to answer. Thank you very much. Any questions? Somebody has to ask a question. I'm going to ask this column one question, this column, that column. Okay, come on. Either you ask me a question or I'm going to ask you a question. Come on. No questions? How many of you are PharmD here? Very good. And how many of you are fourth year? Oh, okay. And you all have studied pharmacology, right? Yes? Okay. What about you? Any questions? Okay, how many are PharmD? Very good. And so when you're PharmD, you are necessarily 50 6 years. Okay, very good. So all 6 years. Okay. How many of you are in B Pharm? Fourth year? Third year? Okay, very good. And is there an opportunity to do PharmD after B Pharm? You can do continuous. 3 years. Okay. 3 years? PharmD. Okay. And how many of you are PharmD? Very good. And how many of you are B Pharm? Third year? Fourth year? Third year. Very good. PharmD? Okay, very good. And B Pharm? Okay, good. Wish you all the best. You can you can somewhere or you can reach out to me. Any questions? Is any way I can I can be of use. So there is a question.
You can ask questions if you have.
Yeah, please ask questions.
Hyperlink.
Clonazepam, yeah. So I think it's always a question. I mean, the question is, clonazepam has a lot of side effects. How come it's still being used? So it's all like that, right? What did Galen? Galen was original Middle Ages scientist. He said all drugs are poisons, right? What determines poison? The dose. So it gives a large dose, but another way to look at it is that it's risk versus benefit side effects. They can still, if their function is improved, then that is good. Schizophrenia drugs are a good example. The patients hate taking it because they don't suffer hallucinations, delusions, but their mind is all like, they're not having clear thought, their movements are hard to do, they hate it. But nevertheless, in terms of the harm they can commit to themselves in terms of having the hallucinations and all, versus not having that and not feeling very good, you can say, okay, this is better. So that's, similarly cancer drugs, right? They're all cytotoxic drugs. But then we are using it because we know that some of them will prolong life or whatever.
Yeah. Is any research is going on to reduce the side effects?
I think from some ways, for example, biologic drugs, although they're quite expensive, they tend to be a lot more selective. The biological drug, let's say if you're an anti-TNF antibody, it doesn't have any other side effects but whatever TNF related side effects only. So in that sense, certain mechanisms tend to avoid a lot of other effects because they are just very clean. However, because they're TNF antagonists, you can get more infections, you can You can latent infection if you're ever exposed to tuberculosis bacteria or some kind of a virus, it might come out because your immune system is now suppressed. So it's a mixed bag. You will always have some side effects, but certain drugs tend to have like more mechanism-based side effects versus other drugs. Like this one, clozapine, has so many pharmacologically, it has so many targets that you don't know what response is for what. Whereas with your effect and your side effect is related. Any other questions? This is just something, even my students, a lot of students also, they don't ask questions. But for you as students, you get most benefit when you're involved. The way to be involved is ask questions. If you're shy of asking questions in a group, go to your professors and ask them one-on-one. Because that is how you show your engagement. When you show your engagement, your brain tends to process information much better. If you sit quietly and you listen to it, go home to class and you read it, it's very hard to retain the information. On the other hand, in class, if you discuss, you ask questions, you talk to your friends about it, then your brain processes a lot better. Your long-term potentiation, those synaptic connections become stronger. So for your own benefit, engage, talk about the stuff, especially if it's of interest to you, valuable, you should always ask a question.
The terms you have used, clean drug, pure
Yes.
drug,
Yeah.
they are not textbook terms.
No, they are not. No, just a bit. By clean, I mean, in fact, in pharmacology, we were taught that no drug is specific. Drugs are selective, non-selective. What does it mean? That means, let's say at 1 micromolar concentration, it's binding to muscarinic receptors. At 10 micron, it's binding to muscarinic and nicotinic. Exactly. So it's selective. Only antibodies tend to be more specific or clean, but it's just a loose term I'm using.
That's when most drugs are dirty.
Dirty, exactly. I agree. It's all we think that an example is, I'm probably old. Our teacher told us that theophylline is a phosphodiesterase inhibitor. That's what we studied in pharmacology textbooks, and that is the reason why it increases cyclic AMP levels, because cyclic AMP hydrolysis is prevented, and cyclic AMP increases and smooth muscle relaxes. Right? That's how theophylline helps. But then subsequently, when I went to graduate school and later, they found that actually at that concentration, theophylline doesn't do anything to cyclic AMP or phosphodiesterase. Instead, it acts on adenosine receptors. So that's again the beauty of science, because at this moment you think it is the thing, but somehow some information comes, you revise your thing. You don't say, oh no, that is what is right, this is not correct. So then your new information comes, you learn new things, and then you change your hypothesis accordingly.
How are the students in US different from Indian students?
So I'll tell you this, because I don't know, because in this case I don't know the students in this class.
You've been
But
an Indian student.
no. By that I mean I can compare with how we were as students, right? So we were Latvia. No, so I'm comparing those two. The cohort that we were when we were students, the other cohort is the students I see there. So there's a time difference also. But we tended to be younger, because we came from high school more or less directly. And so that's one difference. There the students are older. A lot of them may have served in army or armed forces. They're older. They come back and army pays for them, so they go back to school. So they're more mature. And they would have done a lot of things. They would have done BSc, BE or BSc, whatever, and then maybe masters, public health, this and that. And suddenly they say, okay, maybe pharmacy is a good choice for them. So they'll come back. So typically they tend to be more mature. And mature means that also that, and when we were paying, we were, of course, like government, payment was not that an issue, right? These people pay a lot of money, whether it's private college or government college, there they pay a lot of money. And therefore they are they're very serious about it. Most of them, a few will be not serious. Most of them are very, very serious. When you tell them, you teach them well, they think this is your duty. right? If you don't do it, then they will not forgive you. Whereas here, when we were students, sometimes certain professors we used to go and say, yeah, we used to appreciate a lot. We were very grateful. In fact, every time we come here, we visit our teachers and all. We were very grateful. Because there are professors who don't come to class, you go and say, sir, there is a class now, and so they won't come. Like that, that doesn't happen. And the other thing is, our teachers here, when we didn't complete syllabus, they would do special classes to finish syllabus, right? There, they won't do. Everything is scheduled. No class is ever missed, rarely missed. Let's say there is some weather-related thing, there's a lot of snow where I am. When that happens, then the class is cancelled, right? You don't make up that class. You just put a video or something on the thing, they study. Because they, if you say I'll make a class, they won't also come because they are themselves very busy in the evenings, they're working or doing something in a pharmacy, they typically work. So they are not going to come, but because their schedule is all fixed. So in that way, they're very organized. And what else? So these are some differences. I can't think of other things, but there are a lot of some small differences like that.
Major thing is they come to study because they want to study.
Correct, exactly.
Which is not same.
Yes, yeah. So here, some family says go, then you go, right? When I came to pharmacy, I didn't know what pharmacy is. But I came because I got a seat, I came.
They're happy with the last statement.
I think so. Yeah, I think so. I mean, there's not a lot of work done. Now, the thing is, we don't have any new drugs, right? So how do you test that concept, right? This actually can be used to find a new drug and that will go into the market and becomes a drug. That is, I feel like, for example, NMDA antagonists, like I showed you, disrupts it in rodents and animals. NMDA antagonists like ketamine can be disruptive for this response in humans. But the opposite, clozapine improving it, has not been done in humans yet. But I do think that it's very translational. Yes?
21st century, what is the starting point for this study? CAD?
No. CAD is good. And CAD is useful in certain conditions. So the question is CAD useful in drug discovery more in 21st century? What is CAD? CAD is very good, computer-aided drug design. Now, in order for you to use information about the protein, its three-dimensional structure, how this docks and fits in that, all the energies associated with it. For most things, we don't know anything. Like in schizophrenia, what is the target? We don't know. How does it work? We have some hypothesis, we can test something. Now, a beautiful example of CAD and like focused drug design happened with COVID virus, right? Because the COVID virus is like very similar to, I believe, cold virus, and they already had the structure, three-dimensional docking, everything figured. Influenza virus or some related virus. But the bottom line is you had all the information. They just had to do very little data to figure out what COVID's structure is. So when you have all that information, now all you need to do is figure out a molecule that docks that nicely. Within 6 months, you had COVID trials going on. So in those situations when all the information is known, then you can use CAD to make new drugs that dock. There are smart medicinal chemists who can do that and all. But for most disorders, you don't have that background knowledge. Three-dimensional structure, then it's relatively easy, particularly something. On the other hand, if you want to make an agonist, it's very hard. Agonist, then you have to look at what is the natural ligand for it, and then go from there, trial and error, structure-activity relationship. So that's the thing. I think CAD will have a big role, but when we were students, they said combinatorial chemistry is going to come and disrupt everything and all. It didn't happen. It's still today, they still do old-fashioned, okay, this methyl group is active, what will happen if I put an ethyl group, propyl group, and then suddenly it starts going down, then you remove propyl and say keep it to ethyl, like so, ethyl group and so on. So like that, it's still old-fashioned, but there's a lot of other stuff also, and sometimes.
Right now, companies have so many molecules potential.
Yeah.
Simple chemical trial and
Yeah. What they do is For example, my program, I work in a specific, over the course of say 4 or 5 years where we are actively discovering drugs, make every month, let's say they'll make 2 dozen compounds, 3 dozen compounds, whatever, and then they'll do in vitro screening and prioritize some. Now every time they make a new molecule, what they do is they submit it to a like a bank essentially. It's called a library, drug library. So then they submit it. Then you have new drugs, right? Then you say, I'm not working on schizophrenia anymore. I'm working on say antiviral. They will don't care. They will take all those drugs and put in high throughput screening and see what binds. So that way it's very sort of forced. It's not very smart strategy. They'll screen like 1 million, 2 million compounds in their library over 2 months period, and they will come up with some hits, and then they'll take those hits and start making structure activity relationship on those hits. So that's how they use it. So every drug that is made is put in a library, and that library is used to screen. That's the biggest, we used to, when we were in pharmacy, we used to wonder why we are not generating new drugs, whereas they are generating. Because they have this advantage of having libraries. If you start making libraries now, in certain time you will. There is no magic. It's just a process that they follow, based on which they develop drugs. Any other questions? I want to hear some students asking questions too. I think they're all scared of Sudhir sir. If you go out, then they'll ask questions.
They understood everything perfect.
Yeah.
They are very brilliant.
As teachers, we know how that works.
They are very brilliant.
Dear students, ఇద్దరికే డౌట్స్ రావు ఒకరికి 100% అండర్స్టాండింగ్, ఇంకోటి జీరో. నేను అనుకుంటే ఎవరికి జీరో, ఎవరికి 100% ఛాన్స్ లేదు. మీరు భయపడుతూ ఉంటారు ఓపెన్ కావడానికి, డౌట్స్ అడగడానికి. అది భయపడినంత కాలం మీకు ఏం తెలిసినా తెలియకుండా తెలియనట్టే జీరో. ఇప్పుడు ఇక్కడ టీచర్సే మీరు ఇంటరాక్ట్ కావాలి. టీచర్ ఇంటరాక్షన్ ఓకే, కానీ స్టూడెంట్ టీచర్ జరగట్లేదు. ఇట్లాంటి ఆపర్చునిటీస్ రావు. జస్ట్ ఓపెన్.
మీరు పొద్దున జాబ్స్ లో ఇంటర్వ్యూస్ లో అడుగుతారు కదా, మాట్లాడకుండా ఉంటే యూ వోంట్ గెట్ ఎనీథింగ్. సో యూ షుడ్
మాట్లాడకపోతే మీకు ఏం వచ్చినా రాకుండా ఒకటే
కరెక్ట్. దిస్ ఇస్ a risk free opportunity.
మరి ఏం లేదు, ఏం భయపడాల్సిన అవసరం లేదు. ఓకే.
Yeah, I mean robotics are very good at doing very routine things, right? Repetitive things. So for all that library screening I said was robots. So we used to have like it's called lead evaluation or lead discovery, where that library will have many compounds that will recover. Then you connect them to cables and then do it. And it's used just one program after another program and all. Then you can develop a robot do that, right? If they are very specialized things, then there is no point in investing a lot of money for making a robot, so it will do one thing and then it stops. Eventually, we'll probably have robots that we can do many. Right now, it's very repetitive, and those are done for like again you're doing it 100 times every day for like every day of the year. Then you make a robot.
Procedures you develop for this.
No, when we developed this first, we did it. I was in a company. My lab had technicians who would. Now, when I do it in the pharmacy school, I have students who are PharmD students. They're essentially a lot of B.Pharm. Then PharmD students, I have PhD students who do it. Typically, PhD students take the lead and they do mostly. PharmD students observe and they contribute. it's making an incision, cleaning up the skull, disinfecting it, then on the, and then putting an electrode so it sits on top of the dura mat, and it's there, and then you put some dental cement and close it, and then you suture back the skin, and then give some post-surgical or analgesic, let them recover, and after like a week or 2 weeks, you handle them so they're not stressed for a week or 2 weeks. Then you start recording for them, and you do that a few times. Then you start using the pharmacological study. It takes time, but it's not too hard. It's just procedure.
So, and coming out,
Yes.
related to them, as you said, alpha waves, gamma.
Yes.
If I do meditation, which waves?
That's a good question. So it's not clear. I've seen some data people generating more theta, more gamma, and so. I'm not sure if there is anything specific to one band, but there are a lot of studies like that. I remember seeing theta oscillations and gamma oscillations. Good question. Yes.
What is the future of forming and biomarkers or pharmacovigilance?
Yes. So, okay, you said what is the future of PharmD in pharmacovigilance and health informatics? Yeah, I think it's very good. So the one thing is pharmacovigilance, you should have a lot of future no matter where you are, because any new drugs being introduced, pharmacovigilance is basically important, right? You're trying out and then you need to know what is happening, you follow, track it and all. So definitely where the new drugs are being introduced, pharmacovigilance is important. Regarding the health informatics. So I don't know too much about health informatics, but as I understand in the US, a lot of insurance companies, they don't pay for all drugs. You have to show them that there is value in paying for a particular drug, right? If you have clozapine and you have olanzapine and there's a new drug, they say, why should I pay a new drug because it has patent protection, whereas these two drugs are off patent. I can get it from Reddy's or some other place and we give like pennies.
Generics.
Yeah, generics. So they will not pay. Then these people actually build dossiers saying that look, outcomes are a lot better and more attractive. The patient doesn't need to take it for too long. The patient, his cholesterol levels are lower. So long-term health benefits are better here. So you have to that informatics.
Pharmacoeconomics, they have to prove.
Pharmacoeconomics, exactly. So informatics, exactly. Pharmacoeconomics, health outcomes are better with it. So you have to show that. I think informatics probably has part of this, but maybe many other things in terms of looking at what your competitive products are and how your sales are changing from different regions and who your main competitor are, things like that. But I'm not 100% sure what pharmaco informatics is all about. Health informatics. So what do they do? So does anybody have good information about what the health informatics they do? So I'd like to know if you do.
Maybe medical records maintenance. I think online, most likely online jobs system. I think they maintain the medical records data.
Maybe, yeah.
I think so. I think so.
That may be part of informatics as well. Yeah.
When it comes to working, they are shifting to other areas.
Have opportunity to do gate and all, no? Take gate? No, not here, but they can take gate exam.
You put gate the GPAT.
Oh, GPAT, okay.
Same, but it's GPAT.
Okay, so you can take GPAT and do MPharm and all.
PharmD also level.
Ah, right, okay.
They're equal to PG.
I see.
6 years in
Okay.
India.
Coding was for PG here.
Ah, okay. I see.
In India, they don't do MPharm.
Acha.
Need to do.
Okay. Okay.
It's equivalent to PG.
Yeah. So one thing I noticed, I don't know PharmD curriculum here, but in the US it's become very, very clinical now. It used to be that they study a lot of physical pharmacy, pharmaceutics, all that. Now it's all like, clinical pharmacology, patient-centered. The idea, I think, there is that community pharmacies will play more roles in healthcare, taking care of patients, and as a result, they're trying to train themselves to be more healthcare oriented. A good example is before COVID, I didn't think that people would get vaccinations in pharmacies. They would do it in hospitals and dispensaries. Now after COVID, all the vaccinations, routine vaccinations are done in pharmacies. You can still get it in hospitals. You go to your pharmacist, or all the pharmacists, the first year students, they get trained in like give shots and all. So all COVID vaccines, influenza vaccines, and other kind of vaccines that you have to take, they're all there in the pharmacy. Flu shots, all those things. So it's becoming more clinical. And for industry and all, they don't hire that much of they hire pharmacists for doing other work like informatics.
Formulations.
No, even formulations they don't do. They do typically hire like PhDs or MS in pharmaceutics or things like that. Pharmacists they hire in more clinical subjects and things like that, not in manufacturing or R&D. R&D, clinical R&D, yes, not in basic R&D. I was in basic R&D.
Sure.
So,
Yes.
at the beginning of seminar, I'm just listening about family students, then there is no scope of doing B.Pharm.
No, B.Pharm, yes, there is scope. But see, the thing is,
As I'm a student of B.Pharm.
sure, absolutely, very good question. So some of it is our own responsibility in the sense we need to inform ourselves what will we do, right? For example, in India, all the like industry things, they need B.Pharm and M.Pharm. They're not going to particularly, they may take PharmD, I don't know, for certain other things. But if you're going to go in lab and work, it would be a B.Pharm or an M.Pharm, right? Or a PhD. So in that sense, yes, there is scope that way. And similarly, if you do B.Pharm, M.Pharm, and you want to do pharmacovigilance, you can do pharmacovigilance. If you want to do regulatory affairs, you can do regulatory affairs. If you want to do project management in pharmaceutical science, companies, you can do that. But what we as students, what is our responsibility is not only study well in college, but look ahead and see where do you want to go and what can I do to inform myself of what my path is. So it takes a lot of work and it takes actually maturity to think that, okay, what am I going to do tomorrow? What am I going to do next year, right? And you have to start today. See, you have opportunities. All of us here, we are there available. There are so many hundreds of people who are working in industries and all. All you need to do is say, I need this specific help. I think only you can do, nobody else can. People can say general things, but you have to be very specific. What is it that I like? What is it I enjoy doing? What is it that I want to do more? You have to answer those questions to yourself. Then you go to your favorite teacher who can help you. You talk to them. They will help you. They will say, okay, if you're interested in reading and you can process medical information well, maybe you can do some these kind of things. You enjoy working in a lab. Then they'll say, okay, maybe you can do like an internship in an industry. Even if it is free, don't worry, just do it. Get a little bit experience and then leverage that experience in the next interview you have. It's a process. You have to be a partner in your own development. Nobody is going to develop you. You have to develop yourself. Good question. Good question. You're in third year or fourth year? Third year. So you still have time. You have time and you plan. Sometimes you can take a good couple of friends who are also interested in that path, sit together, brainstorm, see where others are going, see who can help you, reach out to them. Any other questions?
మాట్లాడండి.
వెళ్తా మనోళ్ళు చెప్పేసి మాట్లాడితే నేను వెళ్తా మాట్లాడకపోతే నువ్వే వెళ్తావు
నువ్వు రోజు బెదిరిస్తావు, నేను బెదిరిస్తలేను. మాట్లాడమంటాను, నువ్వు మాట్లాడద్దని మాట్లాడమంటాను.
Thank you, sir. Thank you for your valuable session. I welcome Dr. B. Vijay Kumar, sir, for your presence, sir, on this occasion. Please give us a And I also welcome all the students from other different institutions here for this seminar day. Thank you so much. And now I request Dr. Director Sir, Dr. D. Kumar to address the session, sir.
Yeah. Nothing much to Vijay Kumar and Shivarao were classmates. We are both classmates, Jaypal and myself. They are 1 year junior to us in B.Pharm, but later on I became his classmate. Yeah? So 1 year seat informed lo. So then the seat వచ్చింది మళ్ళీ 1 year job చేసి మళ్ళీ again I joined Inform. So Shivarao యుఎస్ఏలో ఉన్నా కానీ చాలామంది ఇండియన్స్ కంటే మోర్ ఇండియన్. ఓకే, సో ఎక్సలెంట్ ఆడిటర్ యాజ్ అ స్టూడెంట్ కూడా. ఇప్పుడు ఐ డోంట్ నో బయట పబ్లిక్ టాక్ ఇస్తారు కానీ అప్పుడు ఇచ్చేవాడు పబ్లిక్ టాక్స్, అంటే మీటింగ్స్ అయినప్పుడు స్పీచెస్. హీ వాజ్ అన్ ఎక్సలెంట్ ఆడిటర్. అమెరికాలో ఉండి ఐ థింక్ హీ హస్ బికమ్ ఎ సిటిజన్ నౌ, ఐ డోంట్ నో, గ్రీన్ కార్డ్ హోల్డర్. బట్ స్టిల్ ఈ దేశం గురించి ఎక్కువ ఆలోచించే వ్యక్తులలో he does lot of activities in India, friends and group of people continuously. He keeps on thinking and working for the. So adoka, and of course he's our good friend, and though he was a junior 1 year, but still we have a connection, good friend. So okay, 2 months back నేను ఎప్పుడైతే కొన్ని సెమినార్స్ పెడదాం అనుకున్నాను, actually I thought online seminar. So I contacted Shivarao to give an online seminar, but fortunately he said he was coming to India, and when he comes to India he visits Warangal. He visits Warangal or Hyderabad, wherever one professor is there. So fortunately that professor is in Warangal now, so he visited Warangal. So ఆయన కలుస్తాడు, ఆయనతో ఒక డే స్పెండ్ చేస్తాడు, వాళ్ళ గురువు మా గురువు. సో ఆయనతో ఒక డే స్పెండ్ చేస్తాడు. సో luckily that professor is in now Warangal. So he is in Warangal, so he came to Warangal, he gave his lecture in Care College. Now he's going to give one more lecture after lunch, another college, and he'll go back. So thank you Shivarao for coming over here. మా హక్కే అది, థ్యాంక్స్ చెప్తున్నాను. సో ఆన్లైన్ అంటే ఆన్లైన్, ఆఫ్లైన్ అంటే ఆఫ్లైన్, ఇట్స్ పాసిబుల్. మీలో చాలా మంది కూడా నిన్న మార్నింగ్ ఒక అబ్బాయి వచ్చాడు, వంశీ అని, మీ సీనియర్, రైట్? ఫార్మ్ డి 2012 అంటే 18 కార్డ్ ఇస్తాను, వెడ్డింగ్ కార్డ్ ఇస్తే కాలేజ్ వచ్చాడు. సో ఈజ్ ఇన్ చికాగో నౌ. సో ఈజ్ వర్కింగ్ ఫర్ ఎ కంపెనీ. సో లక్కీగా నాకు వెంటనే అనదర్ ఫ్రెండ్ ఆఫ్ Shivarao, క్లాస్మేట్ విజయ్ కుమార్ ఈజ్ దేర్. సో ఐ జస్ట్ కాల్డ్ ఇట్. ఏం అవసరం ఉండదు ఎవరికి, నెంబర్ ఎక్స్చేంజ్ అయిపోయింది. ఇద్దరు సేమ్ కంపెనీ వర్క్ చేస్తారు. వాడేమో పెద్ద పొజిషన్ ఉండొచ్చు, ఈ అబ్బాయి కొత్తగా వెళ్ళాడు కదా చిన్న పొజిషన్లో ఉండొచ్చు ఇప్పుడు. సో ఎక్కడో లింక్ చేసుకుంటేనే ఒకటి మాట్లాడమని చెప్పాడు జైపాల్. ఇంకోటి ముఖ్యమైనది ఏంటంటే మనకు కావాల్సింది ఈ కమ్యూనికేషన్ అనేది ఉండాలి మనుషుల మధ్యలో. లింక్ అంటే మాట్లాడుతుంటే వస్తుంది, కమ్యూనికేట్ చేయడం అంటే ఇక్కడ క్లాస్లో మాట్లాడడం కాదు, బయటికి వెళ్ళా కూడా మన ఫ్రెండ్స్ తోటి, మన సీనియర్స్ తోటి, జూనియర్స్ తోటి, బయట సమాజంతో ఉంటే, అంటే అవసరం కొరని చెప్పట్లేదు నేను, ఎప్పుడైనా అవసరం పడవచ్చు పడకపోవచ్చు, బట్ అది ఉందనుకోండి మనం ఏం చేస్తాం, ఛాన్స్ వల్ల అవసరం పడ్డప్పుడే మాట్లాడడానికి ట్రై చేస్తాం. సో అది కాకుండా మనం రెగ్యులర్ టచ్లో ఉంటే ఒకరికొకరికి ఉపయోగపడవచ్చు, హెల్ప్ చేసుకోవచ్చు. సో ఇది ఎనీవే థ్యాంక్స్ వన్స్ అగైన్. రవికృష్ణ Shivarao, please, you would like to give a small. Vijay Kumar and Jaypal, please present the memento and shawl and a book. Memento is a book only. Yeah. So to Shivarao,
We feel privileged and honored to have our guest, Dr. Digawali Shivarao sir. We have want to extend heartfelt thanks for accepting our invitation, sir. Now I request Feldstate sir. Thank you, sir, for giving an important note on neurological disorders in schizophrenia clinic.